Thank you, Nicole. I'll invite our panel moderator, Chris Whelan, to come up to the stage. And is Kasper in the house?
And Carl.
And Carl? Here we go.
Great job.
And as a reminder, we'll also have virtual attendees joining Oliver, Anna, and John. So they should be up on the Zoom. There we are, as if the magic in the back of the room just happens when I ask. Thank you so much. And as another reminder, this is to talk about all of the content that we've heard about from the DiseaseMap session, including the flagship disease project pitches that John described with Anna and Carl as well. And with that, I will turn the panel over to Chris Whelan, to give him a little bio. He's received his PhD in neuroscience and genetics from the Royal College of Surgeons in Ireland, after which he completed a 2-year postdoc at USC, where he founded and led the Enigma Epilepsy Neuroimaging Consortium. He moved to industry in 2016, working at Pfizer and then at Biogen, where he co-founded and chaired the UK Biobank Pharma Proteomics project, which many of us are very, very happy about. He continues to lead the consortium as part of his new role as Director of Neuroscience Data Science at J&J. Chris.
Fantastic. Thank you, Ben. I'm going to start with a meaty one, maybe a complicated one. So large-scale genetics has helped us build a biological map of human health and disease. And by and large, that map remains static. You have the same genetics when you're young and healthy as you do when you're older and ill. And as we're moving from genetics towards taking more dynamic snapshots of human health and disease with proteomics, metabolomics, single cell, particularly single cell, how are we planning to tackle the challenges of tissue-specific effects and temporal effects? I think it was Kasper actually before the meeting pointed out for single cell in particular, it's going to be essentially impossible to model all the different combinations of effects that turn into biological features. So how do we tackle that? Are there machine learning approaches, predictive modeling approaches? I'll start there and leave it open to the whole panel. Nicole?
Maybe I can start.
Sure.
So I guess my comment there would be that it is very important to do both the detailed functional studies and the large-scale surveys, because they're exploring essentially a complementary domain of biology. And I think what we really need to continue to do is to communicate on the respective value, what we're learning, from one to the other, and vice versa. So, of course, if in a simulation study in a controlled environment in cells cannot be done at this population scale, and vice versa. So I think it's just about, in my view, continuing to grow the understanding of what each data layer gives us and how to exploit it. So of course, perhaps thinking about my presentation, we see humans as a less controlled experiment compared to a petri dish, more complex. But at the same time, where you have real-life exposures that can be seen at the level of the organism. And so I think that the idea is to leverage what this continuous process of learning from large scale and then going and testing it and then taking what we are learning in vitro and bringing it back at population scale. So, for instance, some of the value of some of these this population level cohort data is that it can be imputed into other data sets. And so, for me, really the huge if you want to do huge excitement is that ability to ping-pong between this layer of information and learn from that.
Great answer. Any other thoughts from the panel?
I think Ollie actually summed it up quite well when he was talking about in his presentation the multimodal data and the way we can use perhaps some modern sophisticated machine learning methods. In our lab, we've been using non-negative matrix factorization across our single-cell gene expression data to put together these kind of expression factors which are operative across multiple cell types. And we can then ask questions around factor usage and how those, in our case, gene expression factors are differentially used across different individuals, across different cell types, and across time. And you can imagine, as we start to build out from single-cell RNA sequencing and we load in other data types such as ATAC or microbiome data in the case of inflammatory bowel disease, the factors that we are building will become multimodal factors, and then we can ask, because we'll have large cohorts, we'll be able to ask questions around whether those factors are differentially used, let's say, between patients who are on a drug or not on a drug, people who are responding to the drug, not on the drug. And so that way we will be stepping back from that discretization of cell types and individuals rather than letting the data guide us. And we can ask questions over how those factors are used longitudinally over time.
Brilliant. Thanks, Carl. Yeah, excellent. I see on the screen the remote participants have their hands up. We'll start alphabetically with John.
So thanks. I think I completely agree with Nicole response and Carl's. Well, I just want to add that I think to some extent to add on to that is the environment in which these genes and cells are living. So having perspective components to some of these studies is important. Looking at perturbations that are happening in humans, be they disease flares, be they treatment, as well as looking across different disease stages within a given phenotype, I think allows you to look at the trajectory across these different time points or different stages, which I think can add to what was discussed just previously by Nicole and Carl.
Excellent. Yeah, and Oliver?
Yeah, maybe just to add to that, I mean, a lot of us have already said, I just would like maybe to sort of crystallize our thinking that I think in the end, these sort of exposures, that will be a latent, unobserved state of a sample. And we should obviously use all the data to estimate that. And that includes also all the rich site information available. It's not only the molecular data, it's obviously also other environmental variables, homogenizing questionnaire information, maybe using geographic information about life history, maybe extract over time. So we need to combine that all. And then what I think is then the ultimate output is that we will be able to basically estimate this exposure state of a patient based on all the data observed. And there may be variable usage. So for some exposures, molecular information will be extremely important and maybe even more useful in the long run, than tracking life history. For other instances, we obviously will still depend on these variables. And I think these sort of models that would not only put the cell into sort of a latent space or factor, as also Carl was alluding to it, but also the sample. And I think this sort of duality between what is happening to the individual and then what happens to an individual cell, that's something we have to sort of tease apart. And there will also be new method development that will be stimulated and enable that. And I think it's already pretty clear what that's going to look like once we have data that captures this.
Oh, terrific answer. Still lots of work to be done. I'll shift gears a little bit. I wanted to ask Anna, who's online. So we've spent a lot of time thinking about how to operationalize these projects from a sort of mechanical, technical level. But when we think about the human element, I asked this as somebody who sort of did his PhD and postdoc very much in the team science consortium sort of model. How can we continue to protect and support early career researchers within that team science environment, Anna?
Well, good morning, everyone. It's nice to join you. Please forgive me while I slowly wake up. This, I think, is a really important question, and one that I think we can gather experience from the last few years with how we've approached GWAS and the analysis of genetic data. I think that we've learned along the way that we can use the team science approach to really support our trainees and to foster relationships with collaborators that can help with career progression. I think the challenge that we have with this next stage is that the types of experiment that are involved are perhaps a bigger investment in terms of time and can take longer for them to play out. So I think there are important issues for group leaders and supervisors to think around trust with groups and being clear about expectations from the start, because as we amalgamate data, we tend to forget about the individuals who've been responsible for generating it at the start. So I think having a good framework around collaborations. I think in AMP we do a pretty good job of that because we lead from the top, thinking about how we prioritize trainees with the data that they're generating. But when we move outside of our comfort zone into other larger groups, it's going to be important to travel with that ethos.
Great answer. Anything from the panel to add on that? Okay. Good. Well, let's talk about disease maps. So, go ahead. We have a question from the audience. Melissa.
Sorry about that. Thanks, Chris. This is a question. I actually think this could go for each proposal, although I'm going to focus it specifically to Anna. Anna, this is Melissa Miller from Pfizer. Great talk this morning, and thank you for being here so early. You had mentioned interest in longitudinal data for type 2 diabetes. I'm curious to know what kind of longitudinal data are you interested in collecting, and how do you think that will help with your proposal?
Good morning, Melissa. Nice to hear your voice. I think with respect to diabetes, I have an aspirational list of data that I would love to have, but I don't know how easy and feasible it is to collect. If I think selfishly about my own interest in the pancreatic islet, everything that we have collected as a community is kind of an endpoint. It's your ticket out because it's from an organ donor. I think there are some exciting strategies that are coming into play, led by others, of taking samples from living donors. And to me, having a collection of tissues that would give me the sort of charting of islet dysfunction over time that would take me from normal glycemia through pre-diabetes into full frank diabetes would be really illuminating in looking at what actually happens with the breakdown of the communication between the islet cells and changes in expression. It's very aspirational. I don't know how feasible it would be. It's still pretty new getting tissue from living donors, but I think that would be a very valuable resource to have, and build on that, taking tissues from other organs and sites of interest in parallel, again aspirational. That to me would be a mind-blowing data set.
Fantastic. Thanks, Anna. Thanks, Melissa. Excellent. I guess along similar lines, I had a question around disease maps, and the focus of the proposal was IBD, CAD, and type 2 diabetes. So, my background is in neuro, so I'm curious, is that going to be positioned as like V1 of disease maps? Those are the first 3 diseases, and was it based on, unmet need of diseases, or their prevalence, or what were the criteria that went in there? And could we expect to see a second iteration soon that focuses on other, prevalent diseases, Alzheimer's disease, different cancers, etc.? I guess maybe I'll start with John and then open up to the rest of the group for that.
Well, super. As I sort of mentioned in the intro, it sort of grew organically from a number of different proposals that were disease specific, and I think there are numerous sort of opportunities that these 3 diseases present, given what I'd mentioned earlier. Some genetic overlaps, some already some circulating biomarkers that are overlap between the diseases, and having obviously a very strong inflammatory component, such that we can look at not just the circulating immune cells, but be able to compare those to the immune cells that are sort of participating actively in the pathogenesis locally. So, I think it's sort of like a testing ground. If we can do it for these three, we should be able to do it for others going forward. I won't speak for the ICDA leadership, but in the past, this was the idea, is to try and bring these groups together, try and work out a paradigm that obviously will evolve over time, but as a starting point to then hopefully bring in other diseases as well.
That makes a lot of sense. Yeah. Sure.
These three illnesses do represent some very large public health burdens. They traverse a few different areas of medicine, but are obviously not comprehensive. And the neuro question is one that rides high, certainly in my mind, as someone that cares a great deal about psychiatric illness. They have amongst the most well-studied genetics. There's more starting material. There are more loci known, more genes known, more allelic series emerging. There's just a lot more handles on the follow-up functional biological interpretation. For each of them, there is a path to getting tissue, and being able to not only, not have to rely either, in the CAD case, you're dealing a bit more with postmortem stuff, but still there's opportunities to collect specimens from people with those illnesses in a very kind of clear, straightforward, perspective way to facilitate the kinds of genomic profiling activities. And the other thing that I think John really illustrated in the pitch and is important for the conceptualization is just getting to 3 diseases to talk to each other, to engage around what the next round of experiments are, to argue about what the protocol should be, and try and come to some sort of consensus paradigm for taking us through this process. We'd love to do everything in ICDA. We want to work on human health and disease broadly. It's not just going to be narrowed to IBD, type 2 diabetes, and coronary artery disease. But as a starting point, there are just a lot of things that kind of made this seem more feasible and achievable, even though it is already tremendously ambitious to try and pursue such an approach. So that was the sort of positioning. It's not meant to be exclusive. If there are disease communities that are eager to capitalize on similar approaches conceptually and are eager to work together and leverage the potential resources that we're trying to drum up enthusiasm to build for the community writ large, we are extremely welcoming. We would love to have you join, and it should in no way be considered prescriptive, but this seemed like the best place to get started to trailblaze the paradigm for how to go from maps to mechanisms to medicines.
Yeah, makes sense. Very logical place to start. Excellent. Okay, well, sticking on disease maps, Carl, I might ask you, a lot of the work that was proposed by DiseaseMap focuses on ascertaining tissue samples from disease individuals. Yeah. What will the data generated from those samples tell us about disease that we couldn't learn if we only looked at healthy tissues?
Yeah, it's a really good question, and something that we spend a lot of time thinking of in my group, because we have to go to great lengths to get those tissue samples from disease patients. So I mentioned in my talk that the composition of the guts changes during disease, and so we can only access, let's say, tissue-resident myeloid cells if we access tissue from disease patients during active inflammation. So if we want to know what those cells are doing, we have to get them from disease patients. I also think, touching on something that I also mentioned, a lot of focus has been given to disease susceptibility, and not so much attention, unfortunately thus far, has been given to disease progression and drug response. I think if we wanted to understand the biology of drug response, then we're going to need to look at tissue samples from patients who are on those drugs. Yeah. Also, I think if we want to understand something about the disease trajectories and progressions, it's really important that we access disease-relevant tissue so we can follow, in this example that I've been using a lot, let's say the myeloid cells and how they change in number throughout disease course. And I think if we want to be thinking about prediction as well in terms of personalized medicine, it's also very relevant that we access disease-relevant cells from disease tissues, because that's going to help us learn what, let's say in IBD, what remission looks like. And if we ever have ambitions to have approaches which are just built around treat to target, this idea where you kind of where you dose individuals until you achieve a certain biological response, then we're going to need to learn what those signatures of a kind of remission looks like in disease-relevant tissues. So I think there's lots of avenues over and above what we as geneticists may immediately think of, like EQTL mapping, for example, or differential gene expression, where it really is absolutely important that we access disease-relevant tissue.
Fantastic. Yeah, great insight.
Can I ask? Yep. So, Kasper, I have a question for you. If we step back and look a little bit more broadly about what we're trying to accomplish at this meeting in tackling the V2F challenge. There was a meeting over at the Novo Nordisk Foundation yesterday to kick off the NNFC Centre at Broad. Can you say a little bit more about how NNFC at Broad and ICDA are coordinated? Because, of course, the coincidence in timing and location is not a coincidence.
Yeah, no, happy to, of course, and it was great to see a lot of folks from the ICDA community at that meeting on Monday. So we have a lot of leadership who are involved, who have a leg in both activities, Ben, for example, and Cecilia was also there to speak about synergies between ICDA and the Novo Nordisk Foundation Center. So we have organized and aligned the NNF Center very closely to the ICDA goals, but we're really trying to look at it as synergistic to the maps and models that are presented here today, and they really don't overlap. They are really very synergistic with each other. So I think that a lot of the aims, for example, of the NNF Center, and we have other aims as well, but is predictive modeling. So we're trying to figure out, but using foundational cell models, for example, what are the data sets that you would need to make predictive modeling on, for example, gene expression networks and gene regulatory networks, and then to use those hopefully in a generalizable way across other cell types and states. So those are pretty foundational questions that we're trying to answer that are really complementary to many of the other efforts going on here. So we are also thinking very deeply about how do you organize data in a multi-layered, multimodal fashion so that you can sort of make the data sets interoperable. So here we've heard a lot about genomic data types, expression data types, epigenetic data types. But we also want to try and think about how do we integrate proteomic data types, spatial proteomic data sets, imaging data sets, and so forth. So the question then becomes, how do you anchor those together? Oliver talked a lot about that in sort of the genomic space, but how do you actually go to proteomic and imaging data and cell morphology data types and serve that up in a very accessible way for machine learning experts? I think that's a key question, because as Chris was saying, we can't really measure everything. There are going to be so many cell states and cell types and genetic influences and genotypes that having predictive models that can get us 80% of the way is going to be extremely useful. So those are things that we think very closely about. And also, I guess it's a very obvious comment that I'm making here, but these disease areas are so large. The questions that we hear today are just so broad that, I mean, it's a very generous gift from the Novo Nordisk Foundation. We're extremely happy about it. But that's not going to solve all of these problems. These are multi-billion-dollar questions, frankly, let's be honest. So I think it's important to recognize the synergy and not the overlap.
Thanks so much.
That actually segues quite nicely onto my next question, Kasper. Another very broad question for the whole panel, but particularly for you, Nicole. I was very excited by your talk, seeing the first few thousand iPSCs in the UKB, and would love to see that expand further to 50K and beyond. My experiences with Melissa and Mark and others in the UK Biobank Proteomics project has been we can do 60,000 samples quickly, within an 18-month turnaround. And now we're having discussions around, can we scale even bigger? And I guess the broad question for the panel and for you, Nicole, is do we feel confident as a community that as we move away from genomic technologies to non-genomic technologies, that we can continue to follow the path that was set forth by genomics, where studies of tens to hundreds of thousands are now the norm? Or are there going to be new roadblocks for these new technologies?
It's a really important question. Thank you. When we set up doing this pilot, there was no guarantee that we could do it. Right? It was a big gamble, and actually, we learned a lot along the way. We really set off from the start, and that was actually partly through the experience of UK Biobank in designing these large-scale projects. How important it is to have processes that are robust, automated, trackable. We track every sample that comes through the lab. There's just no mistakes there, and having that accumulated experience of how to set up a project, is really important. Then, of course, there is the unknowns about the technology that has never been scaled at that level. So the kind of throughput that I've shown you, 80 samples per day, 400 samples per week, is something that can be achieved now in a relatively small footprint of lab with 4 technicians working at the time and three computational biologists. So the people that I showed were alternating at different times. So it's actually quite achievable. Then what becomes what becomes a little bit more rate limiting is the ability at which you can recruit donors, for instance. And so, again, in our case, having both UK Biobank that had already a recall ongoing, so we could piggyback on that, and ELGH, they really got a team on the ground, the recall, and not to underestimate, we did this during COVID times. It's been quite, it's that added another challenge. So having that kind of engagement, it's hugely important. So, the data looks good, it's stable, it's robust, and so that, then it's a matter of scaling over time, and, it could potentially be expanded in different directions. So the time that we took initially to set up the pipelines and make them robust is really what's now bringing us to the ability to go into production mode and be stable about it.
Fantastic. Carol.
Chris, I have a question for you, if that doesn't break from all the norms. It's for you and Nicole, actually. When we have these large cohorts and we want to, let's say, profile at single-cell resolution their transcriptomes or their proteomes, how do we decide that now is the time to do it, given where the assays and the technologies are? So you were able to profile, I can't remember how many thousands of proteins. 3,000? Yeah. Okay. But maybe you would have liked to have profiled 5,000 proteins. So, and in your case, Nicole, you're talking about 3' single-cell RNA sequencing. Perhaps, if you waited a year, you could do multiome at scale. So how do we decide as a community when the time is right, given the assays that are available, that now is the time to go to things like Biobank and do experiments on the scale of 50,000, 100,000, 250,000?
It's a great question. I'm pausing because I've been asked it before, and I need. There's so many different ways to answer it, particularly around the number of proteins. What I'll say is that the time felt right to at least pilot. Olink is now at 3,000 proteins, and then their biggest competitor, SomaLogic, is 7,000, and there was a healthy scientific debate over which one to do. And we felt, let's move forward with 60,000 in Olink, show that it can be done. And I think that there's going to be a 2 or 3 year period between this initial 60,000 in UKB and maybe doing something even larger. And in that 2 to 3 year period, I think that those technologies are going to expand and get closer to covering the entire proteome. So I guess that's my more direct answer to that. But Nicole, I'd be curious to see what you think as well.
Yes, so I think often these are actually fairly practical external factors that make the decision. So the moment, in our case, we wanted to make sure we could take advantage of the existing recall, because setting up a recall at that level on its own, it would be quite difficult. And of course, in the case of 3', that was the technology that was available at the time on the Connect, and now you could do 5'. And of course, we wanted to use technology that was sufficiently tested and robust, because that's a requirement. Of course, doing ATAC-seq would require a nuclei and a completely different protocol. So, so yes, of course, you can always wait for the next technology, but sometimes you're not going to gain as much as you would by the learning you've done on starting at that time. So I'm sure we'll be mining this data for a long time.
Definitely. I think Oliver might have his hand up as well.
Yeah, I just to add, I think we should, as a community, there's always a trade-off. I think you alluded to the experimental considerations very, very, very nicely. I think there's also an element of whether we have proof of concept that we're able to go from one level of technology to the next. For the single cell field, as an example, we've seen already quite a number of innovations And there's every belief, that if a new technology comes alive, we would at least be able to impute out or tie these data together. That's for an assay like proteomics, a different game, right? Because you have 3,000 or 5,000 proteins, so the others are just not, not observed at this point. But I think this sort of ability to integrate and assure compatibility, that the technology is mature enough to do so, that for the single cell side has been a factor. And I think we've already seen 3 consecutive revisions of single cell RNA-seq, and that seemed very plausible. And I think that's also a good criterion for then pressing the button to really scale up. But I think we have to accept that there will be new technologies arriving, and I think that's true for everything. And we have seen 10 years of rapid evolution in the single cell space, and now I think there's a bit of maturation, at least on the RNA side. And yeah, I think it's something we have to ultimately live with. And as long as we can integrate it with the next generation, it might be okay.
Okay, excellent. Of course, Kasper.
Kind of begs the question of how to quantify when you don't get much more biological signal from doing another assay, right? Yeah. And I don't know that anyone has the answers to that question, but you could argue, you could quantify how many loci are resolving in a disease now. Okay, if we've got 80% of them, does it really make sense to have another proteomic iteration that's 100,000 times more sensitive? So again, I don't think that's a very good quantification metric I just proposed here, but just to give some sort of a view. Yep. And again, the models that, if we can build predictive models based on the data sets that we're trying to generate, it will be possible to extract from those models what are you actually gaining from adding another layer of single-cell attack-seq or whole proteome data that's quantified or spatial proteomics data. Is it giving you something new in the terms of predictive power? Absolutely. So to me, that also kind of, you can flip that around and then say, okay, what's the most efficient data types for getting 80 to 90% of the biological predictions you're looking for, right? So I think that whole arc and universe of questions are going to be important in a lot of these discussions that we're having. And again, no one has the answers, but it's intriguing to think about it.
100% agreed. I believe that we have 1 more minute left, maybe 2, and I'm going to ask a question. I'll be really impressed if it gets answered within 2 minutes because it's kind of controversial. But we've talked about, genetic map, cell map, body map, disease map. So how do we bridge across those different maps? I think it was Anna in her talk mentioned how, a change in gene expression does not necessarily, equate causation. And there's a couple of high-impact studies that have really alluded to that recently. There was a study in Cell 2020 that showed that there's about 5,500 proteins that show tissue-specific enrichment, and most of those, about 3,000, are enriched at the protein level only, not at the RNA level. So, and I guess the question is here, how do we actually map between DNA, RNA, and protein? Do we expect there to be a bridge that will come about, or can we do anything a priori to try to solidify that bridge, if that makes sense? So, difficult question, but curious to know everyone's thoughts.
Maybe I can start.
Of course.
So, I think it is without a doubt that there's a lot of pleiotropy, a lot of not every positive hit will be a functional hit. So we are going through this process of iterative discoveries, and that's they are just faster than so. I think as I think as in the GWAS field, we learned so much from the data accumulated. We'll be able to continue to do that and use this type of data sets, and we now have at least intellectual frameworks, the ability to model networks, to perturb in vivo and in vitro, and so this is, I see actually as a very exciting challenge for the community in the next few years, that ability to just go back to the data we accrued and to plan the perturbation experiments that we now have the tools to do, to be able to continue to refine the extent to which, for instance, our understanding of pleiotropy or cell type specificity translates into the disease mechanism. And biology is complex, and I think we will not always, there will not be a one answer, univocal answer. Loci are different; they act through different mechanisms, and I think, ultimately, we will still need to be able to. We'll still need to do the experiment locus by locus, cell by cell, model by model. But we can at least accelerate our ability to infer more likely pathways through this type of data.
Excellent.
So I'm a huge believer in the power of genetic variation to help us navigate this space. when you have differences, you can do statistical tests. That's very valuable. If you can get fine mapping and co-localization nailed, you can look at dose-response relationships. And I'm also a big fan of George Davey-Smith's kind of recent push on what he's calling triangulation. And I think that concept, that idea that, looking at a question from one perspective, testing a single variant as your hypothesis and as an exposure, and then a single variant as a hypothesis of the outcome, is much, much too narrow. It needs to be, what can we understand about the genetic architecture that's shaping these phenotypes? How can we capitalize on understanding that genetic architecture to then link through the different layers of biological complexity to the endpoint diseases that we're after, and then use, again, that same genetic variation to convince ourselves that the, say, organoid cellular models from iPS-differentiated populations would actually recapitulate biological signal and show the same genetic regulation that we're seeing in the normal tissue and seeing in the disease state. And so, to me, genetics helps hugely with that question. And given its privileged status of something that is fixed at birth in us, accepting somatic mutation, that really does provide a stronger causal anchor than I think a lot of the other kinds of associative things that can come along. But I do recognize I'm just singing the praises of human genetics to an audience of human geneticists. So, but perhaps that's the right thing to be doing at this point in time.
Fantastic. I know we are over time. Any final comments before we wrap up? I'd love to continue this longer, but any last words? Great. I think we'll
Thanks for having
conclude.
us. Yeah, thank you.
Thank you so much.
Great.